Hello everyone. Today we are going to review the process of DNA replication. In order to understand replication, it is important to note that DNA is typically in the form of a double helix with complementary antiparallel strands. It is made up of a sugar phosphate backbone and 4 nitrogenous bases: adenine, guanine, cytosine, and thymine. Sometimes the structure of DNA varies due to self-bonding of a single-stranded DNA, which causes different secondary structures to form. These include the hairpin structure, stem structure, and the complex secondary structure. DNA replication is a semi-conservative process, meaning each strand of the original DNA molecule is used to synthesize a new strand. There are 3 general rules for this process. DNA is replicated from a single-stranded template. Replication requires enzymes, and raw materials are used to synthesize new DNA. In addition, DNA replication has 3 general stages: initiation, elongation, and termination. The first step of DNA replication involves an initiator protein that is bonded to the origin of replication in order to start unwinding the DNA. Then, DNA helicase continues to unwind the strand while DNA gyrase relieves tension with the help of topoisomerase. The next step of replication is elongation, in which single-stranded binding proteins prevent the single strands of DNA from forming complex secondary structures. In order for DNA polymerase to synthesize DNA, primers in the form of RNA are added to specific regions of the DNA to provide a 3' attachment point for polymerase. It is important to note here that DNA polymerase only builds from 5' to 3'. Due to polymerase only building from 5' to 3', there is a leading strand and a lagging strand. The leading strand has continuous synthesis, whereas the lagging strand is synthesized in fragments, known as Okazaki fragments. These are eventually joined together by an enzyme called DNA ligase. The last stage of replication is termination, which we will discuss in lecture soon. Lastly, there are many differences between DNA replication in prokaryotes and eukaryotes. In prokaryotes, replication begins at the single replication origin, as prokaryotes generally have a small amount of DNA. Prokaryotic replication can be bidirectional, where replication goes in both directions from the origin, or it can occur in one direction, which is called rolling circle replication. Rolling circle replication is generally seen in the plasmids of prokaryotes. In eukaryotes, replication takes much longer because there is a lot more genetic material than in prokaryotes. In addition, eukaryotic DNA polymerase is slower than that of prokaryotes. To shorten the amount of time replication takes, eukaryotes have multiple origins of replication on each chromosome. That's all. Thank you for watching.